Welcome to Translating Proteomics, where we explore the science of proteomics and its growing impact on basic biological research, drug development, and more. In this episode, we cover drug development and specifically discuss what makes a good drug target, how we can better personalize drug development, and how proteomics has and will change drug development. And now your hosts, Parag, Malik, and Andreas Humer of Nautilus Biotechnology.
Welcome to Translating Proteomics, a show where we examine the science of proteomics and its impact on everything from basic biology to food security in a modern world and everything in between. My name is Parag Malik, and this is my co-host, Andreas Humer. Andreas has supported thousands of researchers around the world, helping bring the latest mass spectrometry innovations into the world. And my lab has applied those and similar technologies to answer questions in systems biology, cancer progression, and biomarker discovery. Our goal in these conversations is to expose you to all the different aspects of proteomics and prompt you to question assumptions about what might be possible in the future. Also, I'm hoping y'all will have some fun sitting down with us. With that, let me introduce you to our topic for this episode, expanding the druggable universe with proteomics. Drug development is a complex and challenging journey with more failures than successes, high risks, substantial investments, and lengthy timelines. Consider for a moment that 90% of FDA-approved drugs target a protein. And yet today, very few drugs succeed and reach that critical milestone of FDA approval. Only 2 in 10 will ever return the R&D dollars that went into creating them. Biopharma has focused on a few low-hanging fruit targets. And what's left for proteomics is finding that next wave, that next generation of important targets and ways to target them. So our question today is, how will drug development be transformed when we have ubiquitous, inexpensive, and accurate proteomics data? And I'd like to start by asking you, Andreas, what do you think? What do you think when we have this next wave of proteomics?
So I think my thought process is that most of the targets we have chosen so far to drug are targets that are relatively accessible through the analytical tools we had or through observations of phenotypes. Mm-hmm. So we're mainly treating diseases that can easily be interfered with through drugs. And, if I think about the various forms of drugs or modalities that we have, is primarily we treat diseases where a function, there's too much function. Good examples may be inhibitors. Kinase inhibitors are known to be very useful in cancer treatment. But we, for example, cannot have drugs so far that actually create a function within the human body. So I think this is the generation of drugs that we're going to see coming forward, gene therapy, cell therapy. But that also means we have to understand the proteins that are involved in this in a much more intricate details than we do today.
So maybe let's take a step back and think about the phases of drug development.
Yeah.
So phase 1 is find a good target. And in my mind, when you're thinking about good targets and you're thinking about proteomics, you're first looking for targets that are unique to a disease. So, for instance, a protein that is overexpressed on the surface of a cell in a diseased tissue, but isn't there in healthy tissues. You're also looking for something that's specific and isn't found throughout the rest of your body. So you don't want something that's up in a diseased tissue, but also really high in heart or liver or kidney, where it'll lead to a lot of toxicity. And then I guess abundance is a really handy one. If there's a lot of it, then it's easier to target. Are there other things that you think of as, if you think about just that first step of finding the best targets, what are we looking for? And maybe a follow-up question, why haven't we expanded our horizons beyond the set of targets we're looking at today?
It's a good question, but I think we simply don't have, the detailed information that we need today to say, well, this type of, liver cell with this type of particular, protein expression or protein profile is the one that actually, is a good drug target, right? I think we still have not hit that level of granularity of information that we need on the protein level for sure, that allows us to say, yeah, these are the areas of the areas we can develop drugs towards it.
So there's just a gap in information.
I think there's a huge gap in information. I think we have made a lot of progress studying the composition of proteomes, but I think how they're distributed in time and space is probably not as evolved as we needed to have it for strong drug development efforts.
So I guess when one thinks about this, one could say, well, with the tools that we have, we're able to sample enough of the proteome, and one argument is, well, there just aren't any other interesting proteins to be found. The other argument is they're there, we just can't see them.
Yeah, I think what you're referring to is I think what's a little bit of a myopic view of a proteome, which is it's not only absence or presence of a protein, but it is probably the interactions between proteins. It is probably the processing of proteins that ultimately lead to the dysfunction. And I think if we find proteins that are druggable in that sense, then I think we'll have next generation drugs that are probably more efficient because they actually look at the active form of the protein rather than the simple presence or absence of it.
So let me push on that a little bit. So this concept of druggable, this is a term that's been around for a long time. And there are proteins like KRAS, for instance, that people have said this is an undruggable protein. What does that mean? Why would a protein be undruggable? And how would we know when we were discovering the next great target?
Well, I think one area you have already mentioned is basically the fact that if you have a drug that actually does not only bind to the drug target, but it actually binds to other places where it creates a problem, then I think it is a very difficult drug target in the first place. Because we need to remember, right, the drugs we're taking are going to distribute it through our entire organism or the entire body. And finding the specific location where things are dysfunctional, is an additional dimension of complexity. And we're all this is being worked on, right? I mean, there is discussion around how we can shuttle particular targets to particular drugs to particular targets in the body. Thinking all about the modalities that we're developing, where the gene therapy would be very, very specifically targeted to a particular gene. So I think this is all in process. These are just incredibly complex processes that we're trying to interfere with, and that obviously takes a lot of time and effort, right? I think the other part, and that maybe is something you can answer or at least share your thoughts about. We have been treating drugs like, they should work for everybody. And, we're individually very different. Our metabolism is different. And at a certain point, we know that. But how could we possibly change the drug development effort by maybe doing things differently in the development, particularly in the clinical development?
Yeah, that's a really interesting question. I think to date, when we think about personalized medicine, we think about, okay, we want to measure something and then say, okay, you are likely to respond to this drug or you are not likely to respond to this drug. And we're looking at a protein and we're saying, okay, there's more of this or less of this, or it's mutated in some way. And that's the limit of the information that we're using for the most part to decide to personalize a treatment. And I think if we go beyond that, we might be able to say, all right, well, it's not just that you have this mutation. It's not just that you have overexpression of this, but it's that you have this pattern of 10 things. You have this pathway that's hyperactivated. You have this proteoform that is present in this combination of heterogeneity. And so getting more detailed information about the person in their state at that moment might allow us to better target. And so I think that gets to not just what is the treatment, but how are we giving the treatment? Is it maximum tolerated dose, which is how many, certainly cancer therapeutics are given today, or is it this more evolving profile? But if we're going to get to that, we may have to change the way that FDA approvals are done.
Most likely, and I think you're making a good point. When I think about, for example, HIV treatment and how that has evolved, we used to have one inhibitor that would essentially suppress the HIV virus from proliferating. Now we actually have a cocktail, and then we continuously change the treatment as the disease is being suppressed over a lifetime. I can see the same thing happening in cancer, that you start out with a particular drug, your cancer is trying to evade that treatment, and then you follow up with a different type of drug that now arrests the cancer proliferation again. So I think we'll have a lot more dynamic response in drug treatment in the future, particularly if we understand exactly how biological systems respond to drug treatment. I think we sort of ignore that. We call them side effects.
Yes.
And then we just don't do anything about it.
Yeah. So I'd love to actually take a second and go back in time. So you've seen the evolution of this field of proteomics, and this question of how do I predict drug response? How do I define the best drug treatment, the best drug target? It has been a question that we've been trying to study for decades. And so I'd love to hear your perspective on how the evolution of technologies from the early LCQ up to where we are today and where we're going in the future, how has that changed the way that we're doing those experiments? How has it changed the way we're thinking about those experiments?
So I think that's an interesting aspect of this discussion here, because I think up to the point about 20, 25 years ago when proteomics really came about, most scientists were investigating proteins, one at a time. So with the modern tools in proteomics now, we can, certainly investigate the protein of interest, but we can at the same time see most of the other proteins and what they're doing. And often we see that in response to, let's say, a suppression of one protein, we see a number of other proteins coming up that might cause other issues, right? So I think the tools are much more comprehensive. And I think that was certainly the development over the last 20 years in the tools space, that we have been working on creating tools that expand the horizon on what actually can we observe at any point in time in a cellular system or in organisms.
But how does, I guess what I don't understand is if you're measuring 400 proteins or 4,000 proteins or the whole proteome, how does that change the experiments that you do? Or how does that change the way that you think about the experiments that you do?
Oh, it does change it a lot, because you can no longer follow a hypothesis. And typically people refer to these as hypothesis-free experiments.
So I hate that term.
Maybe you have a better one. I'm happy to adopt that. I think you shouldn't do blind experiments just throwing a sample into a tool, right? I think we agree on that. The question is more about, do I intend to observe a particular response, and I set up the experimental tool with it, or do I have an open, field of observation, and then I can see whether things change? And I guess in that sense, these discovery tools have become much more powerful and allow you to look at many, many proteins in parallel, eventually picking up signals that we're not expecting at all.
Yeah. So the extent to which you widen your lens, it allows you to test a wider number of hypotheses concurrently. So versus before, where you could only measure one protein at a time, now when you're looking at thousands, each of those are independent hypotheses. They're also joint hypotheses about interactions between things. And so that's the power that we've gained over time, is the ability to ask a larger number of questions in a smaller number of experiments.
So instead of hypothesis-free, you say it's multiple hypothesis testing?
I'd say it's a discovery experiment,
Okay.
or it's a multiple hypothesis experiment. Absolutely.
Yeah. Okay. The problem still, though, is that it requires some careful planning about some of the controls you need to do at the same time to be able to attribute certain observations, to real events. And so I think that field is probably not very mature. And I think this is why probably isn't yet the backbone of drug discovery today. It's still a tool that's evolving.
So I think that's a really good synopsis of how we got to today. And as the technologies have evolved over the last 20 years, how that's expanded our horizons. Let's think for a moment about tomorrow or 5 years from now, 10 years from now, as we have new technologies. What are the kinds of things we're going to be able to do that we can't do today?
Yeah, I think you can never have enough resolution, on the individual molecule you want to observe, whether that's time resolution or spatial resolution. I think you can never have enough sensitivity. Ideally, you would like to have single molecule sensitivity. Obviously, the dynamic range is also something that is always a challenge, right? How can you observe, very rare event in the absence of many other very high abundant.
So dynamic range is an interesting one because I've heard both sides of this. I've heard, okay, well, that's great that you can measure that rare transcription factor that there are 5 copies of in the cell, but I'm never going to develop a drug against it because there are only 5 copies in a cell. Is there a point where the sensitivity is irrelevant?
Very good question. My answer would be that there's probably biological noise that's not druggable or treatable. Where the biological noise starts and where the biological signal starts, or biological signal starts and where the biological noise ends, I don't know. That might be very dependent on the mechanism. That might be dependent on the protein. And as you mentioned, transcription factors are usually not in the thousands of copies. They're like a few copies.
Yeah.
So you might have to detect very rare events to be able to understand what's going on. So, yeah.
So I guess my answer to that is similar, but a little bit more nuance. So from my perspective, those very rare proteins, if we're able to measure them, so again, we picture the future where we're able to capture the entirety of the proteome at exquisitely high sensitivity, even if we're not drugging those things, if those are the bottleneck, if that's where the regulation is happening, I want to be able to see them so that I can build a chain of causality from that very rare protein to maybe a protein that's 3 hops down the line that is druggable. But if I can't see that protein, then I'll see some changes, but I won't know the mechanism. I won't know where those changes came from. And so it is actually really critical for us in our future world as we're developing our next generation of therapeutics to have as complete a picture as possible.
So I look at this a little bit differently. I think that if you want to use an analogy, you're building a tower from small wooden blocks. And of course, you can spend a lot of time putting them very precisely on top of each other, and you're probably going to, succeed in building a very big tower. But you can also succeed in, sometimes that one of these blocks is a little bit off, and the tower may still be very, very, tall. But of course, it's less stable.
Yes.
And I think these rare events making a system less stable and more dynamic. And I agree with you that some of these events, if they are observable, are probably a good indicator that the system is about to go and change.
Yep.
I just think it's not necessarily, a particular number. It is probably the cumulative effects of many smaller changes that ultimately go over the threshold that then causes a dysfunction that then has to be druggable, right? So I think of it as more of a, a sand pile analogy, right?
Yes.
Rather than, having to observe a mechanistic or having to be able to create a mechanistic perspective.
Yes. So I'd like to come back to talking about what it means for something to be druggable.
Yeah.
And in the future, what does that mean? I think when I think about druggable, or when I used to think about druggable, I used to think that the most druggable things were enzymes. And that what we were doing was we were altering the activity of the enzyme. And so if something wasn't an enzyme, it was a structural protein, for instance, or if it was that it was hard to drug. But as we expand our proteomics toolbox, our concept of what is druggable also expanding?
Probably. I think when you look at more the new drug modalities, there are actually trying to take advantage of structural changes or by bringing simply two molecules closer to each other. And I think the problem that we'll see is that if you go back to the analogy of the blocks, the Putin blocks, right? You would have to develop a drug that essentially avoids that tower from collapsing.
Yes.
And that might sometimes mean you have to replace a structural component. Is that very easy? I don't know. Is it doable? Probably.
No.
I think that's what the problem is. We need to be looking at if we want to have different drugs of modalities.
So I think, I guess from my perspective, there have been a couple key advances that go hand in hand with the measurement that have really expanded the concept of druggability. And as we go forwards, that landscape's going to expand further. So number one is antibody drug conjugates, because they allow you to use something that is distinguishing between healthy and disease to get a general drug into a place. And so whether that particular protein is an enzyme or a structural protein or whatever, it's really just the fact that it's different that makes it a reasonable starting point for a target. I think we've also seen these CRISPR-based things, and then also these cell therapies where we're taking cells out, modifying them, putting them back in. And so we're changing the behavior of our own endogenous systems. And so as we make those changes, the proteome takes on an outsized importance. When we're taking out immune cells, changing them, putting them back in, or other types of cells, stem cells, knowing that we're not going to create cancer by doing that, we're going to need these next generation proteomics tools to understand that therapeutic window very well about what is effective for treating disease, but unlikely to go off the rails and cause damage.
And it's not just the resolution and the sensitivity dynamic range. It will be the massive scale we need to do that. I mean, if we need to really understand, cells as a therapeutics, we'll have to do many, many measurements, and we literally have to, study their composition in a detail that, probably, is undoable with current tools.
Andreas, I'd like to just chat for a second about that point about throughput and how that impacts the way that you again, in the future, how does that change how we do target discovery? If we can measure, most studies today are done on, say, hundreds, maybe outside thousands of measurements, or sets of measurements. What if we reach the point where we can measure hundreds of thousands of individual cells? We can perform thousands, tens of thousands, hundreds of thousands of experiments in order to find a new target.
The way I think about it is it's about information density. If you really want to capture the complexity of a cell, you have to have the information density, the number of data points that allow you to actually see all those changes.
Yeah.
I always take the analogy and say, well, if you take one picture with your iPhone outside of the sky, would you be able to give a better forecast? Probably not. But if you take together hundreds of camera shots, maybe satellite photos, and maybe other measurements, then you can start to predict the weather and you can start understanding weather phenomena. I think we need to have that same information density for proteomics. We need massive amounts of data.
That connects to a topic that we talk about in even more detail, dynamics and time-dependent data and how important it is. So I'm excited to dig into that. I also think that just the way that you think about experiments, when you think about, I can do 1 million experiments, is very different than the way that you think about, I can do 10 to 100. And so the way that people go about doing science, I think, could be fundamentally altered if we are able to measure the entire proteome across millions of experiments readily. And that's the world that I'm excited to move towards.
Yeah, it's probably your unique perspective of a computational scientist. Most scientists start out with a lab book, and a simple experiment in mind. But I agree with you. We have to think more like computational scientists and think about experimental design in the millions of data points, billions of data points.
Yeah, and particularly as we get these new and emerging AI tools, they're going to help us unlock that next level.
Yep.
We're going to wrap here, and I'm going to share just quickly a few themes that I hope you took away from our conversation. First off, how the amazing progress in proteomics tools over the last 2 decades has really changed the way that we think about target discovery and enabled whole new approaches. Also, recognizing that there's a new generation of tools coming, and they will fundamentally alter the way that we think about target discovery, enabling us to attack the druggable proteome in new ways, as well as find novel targets that previously wouldn't have been accessible. And lastly, as we think about, again, moving towards the future and how throughput and scale will further change our ability to think about doing, instead of hundreds of experiments, potentially millions of experiments. Again, changing what is possible in proteomics. Thank you so much for joining us. Look forward to seeing you next time.
We hope you enjoyed the Translating Proteomics podcast, brought to you by Nautilus Biotechnology. To contact us or for further information, please email translatingproteomics@nautilus.bio or scan the QR code on your screen.